ladies and gentlemen here. I would like to start my talk with this award-winning picture on the left here. This picture got award in 2016, saying that this is life, symmetry in life. Arctic fox in northern hemisphere, all of us know that they live in above Arctic Circle, and the red fox lives in the temperate below that. Due to global climate change, warming, red fox has moved into Arctic fox territory, and this is what happens. Now, well, this can be done in the global scenario. Now, what would happen in Indian Himalayan region? To our shock, 5,500 meters, a rhesus monkey in the Trans-Himalayan region of Uttarakhand was recorded last year. Now, rhesus macaque was never recorded above 2,200 meters in the Himalayan region, but it was really surprising for us to see this animal there. Really don't know how it got there, and they're still there, implying that either it could be on its own or it could be human induced. But these are some of the questions that we are trying to answer under the DST NMC program on how wildlife and microflora are getting affected by the climate change in the Indian Himalayan region. Now, this project is being given to us as a task force on wild fauna and microflora. Very strange combination here. And this is a picture from Gangotri area where the snow leopard, the apex predator in the region, and we are looking at a complete array of fauna in the entire area on how things change. Now, the goal of the project is to look at the drivers of change, both anthropogenic and climate driven, and also look at focused investigations on how animals can be used as indicators, how climate change is impacting them in different areas, develop a monitoring system, a decision support system, develop a spatial database so that we can compare the future, and also take climate change scenario analysis and see how animals respond to them, and also microflora, and build capacities. Now, we have laid out a very nice plan, starting from literature review onwards to field surveys, integrating data, also looking at secondary data, developing the interoperable database, getting the field data together, integrating all the reports, scenario building, and getting the comprehensive report on vulnerability indices, and also enhancing the capacity of all the people at WII and elsewhere. For doing the field studies, we have selected 3 basins: Bhagirathi Basin in Uttarakhand, Teesta Basin in Sikkim, and Beas Basin in Himachal Pradesh. Now, these are the 3 sites that we are looking at a basin approach and looking at a selected taxa throughout. Now, we did a hierarchical design sampling wherein we gridded the entire basin by 16 by 16 kilometer grids, then by 4 by 4, 1 by 1, depending upon what animal we are trying to focus. For brown bears, black bears, and snow leopard, the larger grid size would be the one, 16 by 16. For smaller ungulates and other mammals, 4 by 4. And for all other species, smaller ones, birds, reptiles, and amphibians, and fishes, 1 by 1. And for microflora, we are also looking at even much smaller scale of how things are changing. But in the entire elevation gradient, for example, from Deoprayag till international borders in Bhagirathi, and similarly in Beas and in Teesta, we have gridded the areas, and to get fine weather information, because the area does not have detailed monitoring stations, we have deployed data loggers in each of these grids along with various other equipment and camera traps to look at how these areas are being used. Okay. Just a glimpse of the data that we got from the data loggers. The higher elevation seems to be having higher variation in maximum and minimum temperatures, as you see here, about 3,600 meters onwards, and including the relative humidity also is really varying at higher elevations than lower elevations, starting from 500 to 5,000. This is what we're getting. And coming to the different aspects of microflora and fauna, microflora is a very important component. Your bacteria and fungi in the soil do the functioning, and they may get affected by climate change in the future, affecting the biogeochemical cycling in the area. So looking at that, and some insects, some amphibians, galliforms and birds, mammals, fish, and also the human ecology here. Again, different grid size based on what we're trying to look at. So the 16 by 16, and within that the nested 4 by 4, looking at soil sample collections and lichen collections. And these have been collected, and in collaboration with NBRI, ZSI and other institutions, we're trying to look at the microbial communities and what are their functions and how are they being affected. And for this, we have again a flow chart of how we are going to do this. So soil characterization, physical and chemical properties, the communities, soil DNA, and then cloning, the clone separations and phylogenetic analysis. And also looking at microbial functions, enzyme activities, and so on and so forth. Till date, we have got some up to genus level we are going to identify, but work is underway. We are just 2 years old into the project, and it will be taking a lot of time in this area. Also, we are conducting experiments, open top chambers in the alpine regions. This is a very well globally tested methodology of looking at experiments on how climate change would affect alpine vegetation. Now, these open top chambers have been installed in alpine areas, and you can compare the areas inside and outside. So you have a soil thermometer data logger in both inside and outside, as well as a humidity recorder and ambient temperature recorder both inside and outside in the control. And these are the alpine scrub and meadow areas where we are working on, and you can see the difference. Inside the OTC there is a lot of green cover, and outside it is not. It could be a combination of enhanced temperature inside the OTC as well as protection from wind. Now you can also look at how these vary in the green surface area, that is, sorry, you can look at the cover inside OTC and outside and how things change. So definitely there's at least degree increase in the OTC when compared to what's outside. Also lichens are excellent indicators of both pollution as well as they are very slow growing plants, and we can be a very good tool for monitoring changes. Now lichenometry has come of very good use here. We are collaborating with NBRI to look at again how lichen grows inside OTC and outside, and also in our long-term monitoring plots, wherever we have lichens, we are measuring them and keeping them, because you can see how things changed over a period of time. So we have got permanent plots where we have marked some of the lichen and measured them, and these will be used for future monitoring. For soil fauna, that is the microfauna, here we are collaborating with ZSI. Here again the procedures are same, samples being collected, and the animals are being identified, and their role, functional role in the ecosystem are being documented, and we'll be able to get some results in the near future. Coming to the vertebrate fauna, fishes, amphibians, reptiles, birds and mammals, we have teams that look at different methods specific to those species or taxa or the groups, and then sample the areas. We have permanent plots again, and camera traps for mammals and birds, point counts, scanning for animals, trail sampling, using permanent plots and drag netting, aerial netting, and all these methodologies again standardized internationally for different taxa. Now the interesting thing that we have got is, because Himalayas has not been studied very well in the past, and now camera traps have come as a very good tool to look at what kind of animals are there. Now they are giving us very good results. Now you can see on an elevation gradient the species richness, how they change for insects, particularly odonates, the fishes, amphibians, galliforms and birds, and also the mammals. Very interesting patterns are coming up now in these areas. And also we're looking at both elevation ranges also. Now elevation ranges are a very good indicator. If animals start moving up, then we know that there is something that they're responding to, maybe increased temperature or the changes that are happening in the habitat. Now if you see, there are movements both elevation wise as well as lateral movements also, which we call range extensions. Now again, not much data is available, but if you look at 3 species, leopard cat, sambar, which are tropical animals, now they're increasingly moving up and using alpine areas and so on. I'm not saying that they have responded due to climate change, but this is only the things that I'm trying to point out, that these are the animals that need to be focusing and monitoring in future. And monkeys, I've already mentioned how far they have gone. Now, looking at climate change scenario analysis and visualization, we have set up a state-of-the-art visualization and spatial ecology laboratory where work is underway. You can actually see your finished products and visualize how things change, what would happen. And climate change scenarios and projections are underway using an ensemble of 17 models. Baseline and future projections are underway, and change detections are also being made. These are some for the temperature-based changes for the Indian Himalayan states, and these are for the modeled precipitation changes in the Himalayan states. The higher ones are purple and the lower ones are in light blue. And also for the interoperable database, we have compiled 35 databases under 7 themes of topography, disturbance, climate, ecology, several boundaries, layers that have been integrated so that decision support system can be done very easily. One other thing is literature review. Many animals have been reported in the past by hunters, gazetteers, or naturalists and explorers, all that are being used now to see where did they report in the past, 100 years ago, and where are we seeing them now? Because in a short frame of time, we will not be able to see how changes have happened. But now with the historical locations, many of them are yet to be validated, but we can look at reevaluation. Some even species identifications are wrong, but I'm saying that yes, historical data is coming of great use. So for a very large period of, say, for example, amphibians from 1870 to now, we looked at records and seeing how things are changing. Same for the musk deer, old records and new records, so we can say whether there's any movement lateral or altitudinal, and also for snow leopard. We also started a citizen science program called Wildlife Watch in the Indian Himalayan region for all stakeholders, and conducting capacity building programs and some publications. Some highlights I will show before closing my talk. Macaques in unexpected areas like trans-Himalayan cold deserts of Indian Himalaya is something really surprising. We really don't know how they moved in there. These are the elevations I'm saying, 4,500 meters, where even humans don't live. Only they go there occasionally in summers. As I said, leopard, cat and sambar, tropical animals moving higher. We really don't know how and why. And we got also some new mammals being added to the list of Uttarakhand fauna, like wild dog, Tibetan sand fox. Amazing, because if you have not worked in a place and now you have equipment to use like camera traps, you are getting new records. If a mammal like this can be added to a list to a state, imagine small animals. We also added, again, Tibetan argali, an animal found only in Ladakh region of India, is now in Uttarakhand also. And we also added Eurasian lynx last week based on camera trapping. And the most interesting thing is, Uttarakhand in the last 2 years has recorded photo captured tigers and 3 elevations. One in Pithoragarh on snow, one in Madhmeshwar at 4,300 meters, and one in Bhagirathi at 3,000 meters above. And in the same valley, 600 meters elevation above snow leopards. Now, why would tigers move above, higher? We really don't know. And these are not males that are just wandering. These are females. So there are some things that need to be answered. Similarly, areas that are repeatedly being used by common leopard and snow leopard in the same area, indicating some overlaps in these regions. New records of several damselflies and dragonflies are happening. Some even new to science, maybe, we are not sure. Genetic studies are underway. Also, maybe amphibians like amolops. We are getting a new one which doesn't resemble anything closer to even the Chinese species nearby. And so finally, our progress of work for the next 3 years would be to get all this information baselines standardized for Sikkim, that is Tista Valley, Beas and Bhagirathi, and look at some species, how they respond to climate change. And the experiments that we're doing for microflora will give us some more lights on how climate change is impacting microflora and fauna. With this, I would like to thank all the funding agency collaborators and all. And this project is being carried out at WIAP with a very large team, and we hope to get some good results in the near future. And thank you, audience, for a very patient listening to my talk. Thank you very much.
Thank you very much. If you have any questions to ask, we will have time for one question. art